Hello and welcome to Science Research Weekly, Season 5, Episode 12, Dropping the PepHammer. Get ready for ancient soft sponges, astronomical biosignature spotting via grouped planets, the epidemiology of AI, crop management via smartphone, an examination of NIH funding outputs, dropping the PepHammer, asking if AI agents save time for scientists, and a grant on specialty crops. This will cover material from April 10th to April 16th. Let's get started with our research articles. First up is a late-Ediacaran crown group sponge animal. So sponges, most basal metazoan phyla, and they're expected to have diverged and come out in the Neoproterozoic, but researchers, paleontologists really haven't been able to find them before the Cambrian period. And one of the possible reasons is that the early sponges didn't have biomineralization. And so there's an Ediacaran fossil that researchers just found, Heliocolocylus cantori, which I think actually is a sponge with an organic skeleton, so no speculation, and that would sort of comport with the proposed origins of the sponge, that they actually started out without having biomineralization, which is why they're hard to find pre-Cambrian. All right, next up, we have Perlene drives mitochondrial DNA nucleoid distribution. So mitochondria, they have their own DNA, and they're packaged into things called nucleoids, and they actually need to be spaced across the organelle. It wasn't clear how they actually did this, but it turns out that they actually might do this through this physical process called purling, where it transfers the mitochondrion from a tubule to connecting beads, and then able to essentially distribute the DNA properly. All right, we have this article, P21 plus, TREM2 plus, sentient macrophages fuel inflammaging and metabolic dysfunction, steatotic liver disease. So cellular senescent, so that's when cells get old, and some even call them zombie cells, where they are almost dead but not quite dead. But they're certainly an issue in aging for inflammation. And in this paper, researchers found that some of the sentient macrophages had a certain profile. They could look at them, and they found that in mouse models, they had issues with the liver, and if they took them out, they targeted those macrophages, took them out, and actually reduced liver inflammation without any changes in things like diet. So very interesting. All right, we'll switch over to the Astrophysics Journal, or Astrophysical Journal. And this article is an agnostic biosignature based on modeling panspermia and terraforming. So one of the reasons why scientists, astronomers get excited about exoplanets is that maybe those planets have life on them. And one of the things you can do is look with the chemical signatures of the atmosphere for biosignatures, potential indicators that there's organic life there. However, there's a problem. There can be a lot of false positives. And so one of the ways, instead of just looking at one biosignature, is to actually look at planet level. Instead of planet level, look at star system level. That if you found sort of statistically correlated biosignatures across multiple planets, that would suggest that there's life and it's colonized multiple planets. So it's kind of, I hadn't thought about that before. I mean, certainly this would only be useful, as the title says, things like terraforming would only be useful for detecting biosignatures of significantly complicated intelligent life that can go off their planet of origin and terraform. This would not find simple microbial life, which is probably the most common if there is life elsewhere in the universe, but certainly a unique way of looking at finding biosignatures and finding stronger proof for biosignatures against false positives. All right, that ends our normal article time. Let's dive into the preprints. We've got 3 from arXiv. First, Visplot, a visibility plot and observation and scheduling tool for astronomical observatories. So yeah, it's a free open source web-based tool for hardware visibility analysis and heuristic scheduling, so you can better schedule and look at like observations. So I think the table here, see if we can rotate that. There we go. So you can see some of what that looks like. You can look at a full night schedule example for a set of targets. So that might be helpful for observatories. Okay, next there's AI-assisted writing and reorganization of scientific knowledge. So they looked at the fact that a lot of scientists use generative AI, especially for building upon prior knowledge, so literature reviews, and so they actually analyze articles, et cetera, et cetera. And what they kind of found was, AI-assisted writing was positively correlated or associated with citation disruption, but it was more narrowly, so it was relatively narrow knowledge inputs, as opposed to, I think, broader sort of increase in citation. Yeah, so it's associated with more disrupted citation structures without a corresponding expansion in cross-field recombination. So AI-assisted writing does not broaden the search space of science, but it could have new forms of recombination built from a fairly narrow knowledge input. All right, and one more on AI here. The epidemiology of artificial intelligence. I thought this was a take on it that I've not seen before. Looking at, so you have epidemiological models for things like health and disease, and you can add all sorts of covariates that might predict health outcomes. And they're looking at a variable they called ambient AI exposure. So how much exposure people actually have to AI, both sort of personal, but also more broadly. So ambient is like, yeah, that the AI things that affect populations regardless of visual choice. This would be something like air pollution in a more common epidemiological model, whereas personal AI exposure is direct volitional use. That'd be like smoking. Both are sort of exposures, and there's different levels. So the idea of, a lot of people will use AI to diagnose or look up health things, so that might have an effect on outcomes. And so that was a impact of AI on healthcare that I hadn't thought of, and so I think it's an interesting topic. All right, we will shift over to bioRxiv. First, we have the Common Fund data ecosystem. So the Common Fund is, so NIH has a bunch of Common Fund programs for discovery and integrative analysis, so data resources. And so it's kind of hard to get at. So the authors here create a Common Fund data ecosystem to help bring that together so people actually access and use that data. So I think that's an important service here. Okay, then there's image-based spade values and chlorophyll measurement using a mobile phone, enabling accessible and sustainable crop management. See, one of the ways to manage crops is to make sure you can tell how they're doing by testing them, by imaging them somehow. For example, using color cards, assays, meters, or even things like phone apps. And they were comparing that and seeing how you could do that with just phone apps. It looks like there's varying ability. That seems like whether there's software or hardware, it's fairly fast, fairly easy to use, very low cost, very good on the field, and then depending on what you do here, the precision can be low to moderate. So there are certainly trade-offs. Okay, what we have here is comparing the outputs of intramural and extramural grants funded by the National Institutes of Health. So the researchers looked at intramural grants that NIH sort of funds their own scientists, and then extramural is, individual labs, and they found differences in cost effectiveness. Extramural were more cost effective in common academic evaluation outputs like publications and citations, while intramural were more cost effective for generating research that influences future clinical work, so more closely in line with the agency's health goals. So saw a difference in outputs. Okay, final one, PepHammer, a lightweight web-based tool for bioactive peptide matching and identification. Peptides, those are essentially parts of proteins or very small proteins, maybe how you put it. They're made of amino acids and their potential for therapeutic agents. And it's hard to sort of figure out what their chemical relevance is because it's hard to match and identify them. So researchers created this PepHammer, web-based tool for bioactive peptide matching identification. You can input a bunch of peptides and compare them against databases of peptides with predicted or experimentally validated bioactivities and all sorts of different metrics. So that should be useful for peptide research. All right, we will switch gears here to some research tools. First up, from our bloggers, we have 4 of them, I think, today. The first our bloggers from Stephen Royal, My Domain, proteome-wide scanning for TMDs, transmembrane domains. So he used R to answer the questions, how many proteins in the human proteome have transmembrane domains? And of those, how many have 1 or 2 or N transmembrane domains? So he wasn't be able to find it himself, so he did R to do it for him, or to do it by himself because no one else did it, and looks like he was able to do that fairly successfully. Did it for model organisms, and he has codes here if you're interested as well. And I think that's a great example of just using R to answer questions that don't have answers yet. All right, next on a statistician's R notebook, why most time series models fail before they start. I don't do much time series analysis, but I kind of understand it a little bit. It turns out you can run the model and they can fail because they violate a assumption called stationarity. And so this kind of goes through what stationarity is and how to look for it and make sure your time series model doesn't fail before it starts. All right, next up we have from Seascape Models, do AI coding agents save scientists time? That would be a big thing. We have AI models, but do they actually really help you write faster or better? And so I think this short article, I think it was really interesting. And one of the things that kind of points out the answer is sort of context dependent. And that you might save time on some way, but if sort of trying to decode or debug can actually take longer in the long run. So it depends on how you use them. They're certainly tools. They're not a silver bullet. I guess that's what I took from this short article. All right, and finally, from the Jumping Rivers blog, using R to teach R, lessons from software development. Jumping Rivers blog celebrated 10-year anniversary and talks about some lessons learned, and so of how they actually can teach and do training courses. And so I think this was a helpful article just for overall, as they say at the end, the take-home lessons from this blog are applicable to any big coding project. Doing things like trying to avoid duplication, utilize certain workflows, and focus on what matters by automating as much of the process as possible. All right, with that, some new R CRAN packages. We have GLM4, fitting generalized linear models using sparse metrics, Tiny Rocks, minimal R documentation generator, Baseverse, modern base R functions, and TidyFun, tidy functional data wrangling and visualization. Okay, closing out here, we have some potential grants for you. First, from the National Institute of Food and Agriculture, we have Specialty Crop Research Initiative. So, research initiative to look at challenges of national, regional, multi-state importance for food and agriculture, especially specialty crops. So this isn't just general crops, specialty crops. And I had to look that up, make sure I understand what specialty crops were. So there are things like fruit and tree nuts. Those are certain vegetables, culinary herbs and spices, medical herbs, which I thought was interesting, especially for sort of medical use, horticulture, annual bedding plants, potted flowering plants, herbaceous perennials, cut flowers, cut cultivated greens and foliage plants, Christmas trees, deciduous flowering trees, broadleaf evergreens, deciduous shade trees, conifers, shrubs, ineligible commodity. Okay, yeah, so those are all. So it turns out a lot of crops are specialty crops, but the ones that aren't specialty crops are the sort of main crops, the big crops: alfalfa, peanuts, corn, rice, clover, etc. Eggs, even, I guess, flax. So if you're doing something more niche, you could apply for this grant. All right, switching over to Pharma Foundation. They have a translational medicine postdoctoral fellowship, 60,000 a year for 2 years. And so that's for individuals engaged in multidisciplinary collaborative research training program at an accredited U.S. university. That will extend their credentials into traditional medicine. So there we go. And then finally, there is from the ACS Science Cafe Mini Grant for a relaxed open venue for known scientists and scientists to discuss current topics. It was very small. It's like $500 for local sections to host these cafes. With that, thanks for joining me for Science Research Weekly. Science on.